I'm Mr. Leung. In this video, we are going to go through the Chapter 2 assignment, and this video will be divided in two parts. Part 1, I will talk about the MC questions, and for the Part 2, I shall talk about the long question. So for the question 1 of the MC, actually it's not really related to the cell structure, because this part I shall spend more time on Chapter 5, okay, the human diet. Because for this part you have already learned part of it, okay, for the carbohydrates, for the proteins, for the water, for the minerals, okay, that's something you have learned it before, so I will not spend too much time on it. So for this question, I just want to capture some important ideas for you. So which of the following is a function of the cellulose in plants? So for this part, I would like to capture this part from the book, okay, because you can actually find the answer from the book, okay. So for the carbohydrate, it contains carbon, hydrogen and oxygen atoms. It's just like the fat, okay. So that's why in this part I would like to mention that carbohydrates and fats, they also contain carbon, hydrogen and oxygen atoms. However, the most important thing I would like to point out is that for the carbohydrates, the hydrogen to oxygen ratio in carbohydrate is 2:1, because for the carbohydrates, it contains carbon and what we call water. Water is H2O. H2O, that means the hydrogen to oxygen ratio is 2:1. Okay, so that's H2O, water. Okay, so that's for the carbohydrates. Okay, the difference between carbohydrates and fat. And for the fat, okay, the H2O ratio is higher than 2.1. Okay, that's something I would like to mention here first. Okay, and we shall talk more about it in details in Chapter 5, Human Diet. And you can actually find the answer, okay, in the textbook. Cellulose is the main component of plant cell wall. So actually, I think you can tell me the answer. And I would like to mention one thing is that there are different forms of carbohydrate in plants, and this part will be discussed in different topics. For example, when we talk about the photosynthesis or the plant gases exchange, we shall talk about the photosynthesis. So for photosynthesis is that for the green leaves or the green stem, okay, any green parts in the plant, so they can produce the glucose by photosynthesis. So we can find glucose here. And for the cellulose, we can find in any plant cells, okay. All plant cells, they have the cell wall, okay, as the structural component, okay, as the of the plant cells. And also the starch, it is a kind of food storage, okay. We can find it in the fruit, we can find it in the root, okay. So especially when we talk about the plant transportation and also the plant reproduction, okay, we need to mention that the starch will be stored in somewhere, okay. For example, in the apple or in the banana, okay, that's the fruit, okay. And also the plant, okay, when they are growing, okay, for example, in winter, all the leaves, okay, they fall, okay, all the leaves, they fall. So the plants, they still need food. How can they extract the food, okay? So they extract the food from the root, okay? So that's something I would like to talk about in question 1. And for question 2, it's talking about how can we use the microscope to zoom in or zoom out, okay? So it is the cross-section of a stem, okay, a plant structure, okay, under the different magnification, okay, diagram 1 and diagram 2, okay? And according to the question, we know that actually we are switching from diagram 1 to diagram 2. Okay, so the first question, okay, you need to recall, you need to remind yourself is that it's from diagram 1 to 2, okay, is the processing zooming in or zooming out, okay? Of course, it must be zooming in, right? Okay, from 1 to 2, zooming in, okay? So that's why you need to be very careful when you are reading the question, because once the question is changed, okay, when it is talking about from diagram 2, okay, to diagram 1, okay, it will become zooming out, okay? But now it is zooming in, and the next step you need to think about is that which type of magnification power we should use, okay? When we are using zooming in, we should use higher magnification power or lower magnification power, okay? So that's the question. So zooming in or zooming out. Now we are going to zoom in, okay? And for the second question is that which part are you zooming? Are you zooming in, okay? So where is the part you want to observe? So for this part, I want you to take a look at the green circles, okay? So for this part, I circle it for you. From the diagram 1, okay, actually, we put the whole stem, okay, the cross section of the stem in the middle of our field, okay, under the view, right? Okay, but for the diagram 2, actually, we are not looking at the middle of the stem. We are looking at the peripheral region of the stem. So that's why we need to think about, okay, what do we need to do to make sure that we can observe the peripheral region of the stem. So that's why for this question, okay, we must we ensure that answer C and answer D, either one of them, it is the answer, okay, but A and B, they must be wrong, okay, because the first step we need to do should be to adjust the position of the section on the stage and make sure that when we are zooming in, we can still find it, okay, we can still find it. But the question is, so C or D, which one is the answer? Okay, so you need to recall what you have done in the laboratory last week. Okay, so when you are handling the microscope, okay, to zoom in different things, maybe the cells, maybe the cotton wool, maybe the newspaper. Okay, so what did you do first? Okay, you chose the suitable objective first, or you do the focusing first. Okay, how can you do the focusing? Okay, so we rotate the adjustment knob. Okay, no matter it is a coarse adjustment knob or fine adjustment knob. Okay, for focusing. Okay, so which step, okay, you do it first. Okay, so it will determine it will be 3-1-2 or 3-2-1. Okay, so for the first step, turn the objective. Okay, of higher magnification. Okay, so that means we use high power. Yes, it makes sense. Okay, and the second one, turn the coarse or fine adjustment knob. Okay, so which one you do first? You tell me, C or D, which one is answer? Okay, and this question, okay, somehow because we are doing high power, high magnification power. Okay, so if we would like to make it in details, okay, or accurately, somehow we know that, okay, we remember that, okay, when we are using high power, okay, we should use the fine adjustment knob for the final focusing. Okay, but for the coarse adjustment knob, okay, we are moving the whole body tube, okay, to try to make the slide, okay, close enough to the objective, okay, for your observation. So C or D, which one is answer? You tell me. And for question 3 is talking about the light microscope. Okay, so any inspiration, okay, is developed after Robert, okay, use a light microscope to observe the cells. Okay, so somehow you can find some information, okay, from the book. Okay, so Robert used the light microscope to observe the thin slide of cork. Okay, and finally somehow he was observing the cell wall. Actually, okay, that's something what we call cell wall actually, but not the actually the cells. Okay, but after that, actually other scientists, they use the result, okay, or the conclusion, okay, from Robert, okay, to develop, okay, the idea of cell theory. All organisms are made up of one or more cells, okay, and the cells is the basic unit of the life, blah 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 blah. Okay, so that's the concept, okay, of cell theory. So that's why somehow B is the answer already. But I would like to spend some time on explaining why A, C, D, they are wrong. Okay, so A, D, they are in one group, so I highlight it in red color, okay, and C is the other group, so I highlight it in green color. So option A and option D, why they are wrong? Because for the virus, because for the fine structure of the organelles, okay, they are just too tiny to be observed under light microscope. Okay, so they only can be observed under electron microscope. Okay, so that's why for this question we are talking about light microscope only. So that's why A and D, okay, is out of the scope, okay, observed by the light microscope. So A and D are wrong. Okay, but for option C, okay, although bacteria can be seen under light microscope, but we have to take a look, okay, very carefully at option C. Option C is talking about the discovery of bacteria as a disease-causing agent. Okay, yes, in fact, we can observe bacteria under light microscope, but actually we cannot confirm that all by simply looking at the bacteria, and then I know that, this bacteria, it can cause disease, and this bacteria, it cannot cause disease. We don't know that. We just find them. They are bacteria. Okay, but what are they? Okay, will they cause any disease? We don't know. We just find it. Okay, but we don't know whether they can cause disease. Okay, so that's why we need to do some further process. Okay, further steps to make sure they will or they will not cause disease. For example, put the bacteria, okay, maybe in any other animal, okay, put the bacteria in the chicken, okay, and then let's see whether will they suffer from any type of disease, something like this. Okay, so for option C, by simply looking at the light microscope, okay, to observe the bacteria, it cannot tell whether they will cause disease. So that's why option C is also wrong. Okay. And for question 6, I don't have much to to talk about it, because you can find the diagram, okay, in the book, okay, and especially focus on the top right hand corner. Okay, what is the relationship between cells and the tissues? Okay, similar cells group together to form a tissue. They work together to perform one or more particular functions. Okay, so by using this, I think you can tell me what is the definition, okay, or to describe what is tissues. Okay, and number 7 is talking about the difference, okay, between prokaryotic cells and eukaryotic cells actually. Okay, so which of the following structure are found in prokaryotic cells? So this table I extracted from the book, and I also talk about it in the lesson to mention that when we are doing revision of the comparison between prokaryotic cells and eukaryotic cells, the first priority, okay, we need to talk about is the true nucleus. Okay, whether they have or they don't have the true nucleus. For the prokaryotic cells, they do not. Okay, they absent. Okay, the true nucleus is absent. But for the eukaryotic cells, okay, they have true nucleus. Okay, and because we can observe the nuclear membrane surrounding the genetic material, so that is the true nucleus. Okay, and this is the first step. And the second step is that if they have the nuclear membrane, we know that there will be extended membrane bounded set to form the ER, the endoplasmic reticulum. So that's why for the eukaryotic cells, because they have the true nucleus, they also have the endoplasmic reticulum. Okay, and these two structures, okay, they are also absent in prokaryotic cells. Okay, so that's why for the question, okay, option B must be wrong, right? Okay, there is no ER in prokaryotic cells. Okay, and for the membrane-bounded organelles, okay, in prokaryotic cells they are also absent. But for the eukaryotic cells, okay, they have such membrane-bounded organelles, double membrane organelles. Okay, for example, mitochondria and corpus. Okay, and I wanted to mention one thing. Okay, look at this arrow. Okay, this arrow is trying to clarify the concept. Okay, because some students, they may misunderstand that. Although, okay, they may misunderstand that the prokaryotic cells, because they do not have mitochondria, they do not have such membrane-bounded organelles. Oh, mitochondria. What is the function of mitochondria? Oh, is to carrying out respiration. So that's why prokaryotic cells, they cannot carry out respiration. Okay, it doesn't make sense. So take a look here. Although the prokaryotic cells, okay, they do not have mitochondria, there are still some structure carrying out respiration, because respiration is one of the 7 characteristics of the living organisms. So that's why even they do not have such membrane-bounded organelles, they still have the structure, the enzymes, to carry out such very important process in the cells. Okay, so that's why even they do not have mitochondria, even they do not have corpus, they are still able to carry out respiration and photosynthesis, right? Okay, and for the last part, it is about the ribosome. Okay, so for the ribosome, according to the table, we know that no matter for the prokaryotic cells and eukaryotic cells, they also contain ribosome. Okay, so the question is not about yes or no, presence or absent. It's talking about where are they, or the size of the ribosome. Okay, so for the prokaryotic cells, actually the ribosomes are smaller and they are lying freely in the cytoplasm. Okay, they are free floating. Okay, but for the eukaryotic cell, because they have the true nucleus, they have the nuclear membrane, they have the extended nuclear membrane. Okay, so the ER, and that's why some of the ribosomes, they can attach to the ER, okay, and some of them they are lying freely in the cytoplasm. So that's why actually both of them, both of prokaryotic cells and eukaryotic cells, they contain ribosome, okay. And for the question is that the following structures, which one of them is found in the prokaryotic cell? So actually A, B, C are wrong. So the final answer must be D, ribosome. So I would like to mention that the very, very important concept, okay, you need to clarify, you need to understand is this part, okay. Although, okay, they do not have the membrane-bounded organelles, they can still perform the important process, okay, in the cells, okay. Respiration, photosynthesis, something like this, okay. So that's for the part 1. So you can switch to the part 2 to take a look at the long questions.